we're looking at one of those little squares. So what are those little squares called? C-squares, yeah. To here, to the very beginning, you've got the volume written down there. So you know the volume of a C-square. No, not too far. So if this was your foot, you need a back. Okay, so the title of this investigation is, so if you just write this on top of a piece of file paper, experiment 3, sorry. Investigating yeast population growth and we'll just give a little bit of background information here. So I made up a yeast suspension, which is basically yeast and water. And I divided it into two, and I put one in, I'll write this up, but I basically put one into a beaker that had a 1 gram of glucose added, and the other one had 2 grams of glucose. So equal volumes. And then I've run out of space here. You're just writing on file paper, so you can say after 24 hours, samples were taken from each flask. So which flask would you expect to have a higher number of yeast cells? Jasmine? 2. Okay. So, and whenever we looked at this, everybody agreed that the sample that had the 2 grams of glucose had certainly looked as if it had more yeast cells in it. So you need to decide, if you can't remember, you need to decide which sample is flask 1 and which sample is flask 2. Okay? Then you need to pick one of these squares here. Now, I'm just going to take this at random. There's a few clumps, so there's a clump there, which we're going to avoid that because that's an issue later on. I mean, in an exam, practical exam, they might say, what are the difficulties or how do you prevent clumping? Just make sure you stir it, or make sure that you stir your solution well, because it's very hard to count the number of cells there accurately. So we're going to don't pick a square that has got a clump in it. But for example, pick a square. So if I pick this square here, if there you can see 3 lines there. In that case, you take the middle line. Okay. So basically, you just outline your square. Now you need to be careful how you draw that. And then you count your cells. So if I was counting that cell, that would be 1, 2, 3, 4, 5, totally 6, totally inside. Count the ones at the top. So what I say, that was 6, 1, 2, 3, 4, 5, 6, 7, 8, and the left, 9, 10. Oh, one more. I think I would count that one, but not the one at the bottom there. So it'd actually be 11 then. So what you want to do, you want to go around a type C square. So choose wisely. Choose one that you can, that don't choose one with a big clump or one that's got one. Don't choose one that's got a cell like that touching the bottom and the left. So remember the rules. Totally inside or touching the top and left. Ignore bottom and right. Penny, I can't see squares. I can just see horizontal lines there. Can you see vertical lines in your photograph? If you can see vertical lines, you're going to have to just go over them in a pen or something then. All right, because I can't see them on that. Just be careful, because whenever you look at the grid, if you take your photograph there, you're only going to have vertical lines, or if you take it there, you're going to have horizontal lines. You need to make sure that you should have taken it there. Okay. If you don't think you've got a decent photograph, then get somebody else's. All right. So, on your 2 photographs, if you need to cut them out, or if they're on separate things and glue them in, that's fine. If they're on just a sheet, that is also fine. But you need to label. There's glue here if you need it. So what you need to do So either cut your photograph out, stick it in, or at the side of your photograph. It doesn't take much space, but you need to show, you basically need to pick a square, a type C square as an example, and show that you can count the number of cells accurately. And you need to do that on flask 1 and flask 2. Okay, so do it on both of those. Okay, then you need graph here. So actually Penny has got a lot of C squares, so you need to get 10 figures written down. So if you've got 10 squares in your photograph, that's grand. If you haven't, get a few and then look at somebody else's photograph and count theirs, all right? So you need then to produce a table with 10 results for each flask. Now, I can't give you a results table. That wouldn't be ethical. It wouldn't be moral. I can't give you a table and just say, copy this down and write it. But think, you've got 2 sets of figures. You need to say that, you know, where they're from. I would include the mass of sugar as well in your table somewhere, just to make it easy. So remember, flask 1 hasn't got as much sugar as flask 2. So make sure that you are looking at the right sample, that you're getting, on average, higher figures from flask 2. Now, you don't need to say, like, experiment 1 to, or, you know, say, you don't need to write down, you know, in that example that we used yesterday, the squares were like numbered and you know, like it was A3, B2. You don't need to do that. You just need 10 figures written down and then put an average or mean at the bottom. In terms of units, what are we counting? We're counting the number of yeast cells per what? What are we counting them in? Penny?
Per square.
What square though? What type of square? What do we call that? So if we think back here, table. And your table has to have okay, so the 2 flasks, 10 numbers per flask, and there was 10 numbers of cells. You need a mean at the bottom, or average. Remember that you're counting the number of yeast, so yeast has to appear somewhere in your title, and it's type C squares of a haemocytometer. So you need table with headings, and a decent title. Sometimes that's called a caption. You need to mention the living organism. You need to mention the fact that we've got 2 flasks. Somewhere in that you need to have the amount of sugar. And your numbers are number of yeast cells per C square. So you can get to work on that. All right, so I'll let you show a type C square from each photograph. If you've got 10 squares, that's good. If you haven't, then you can go around and look at somebody else's photograph and take some numbers from that. Okay, so once you've got your averages, you will end up with a mean number of yeast cells per C-square for flask 1 and flask 2. So folks, if we just put our pencils down, I know you might not be at that stage right now, but I'm going to explain what you're going to do, and then you'll finish this off at home, bring it in on Monday, and hopefully then everything will be done and dusted, and then we can put this to bed, this practical to bed. Okay, so you've got your photographs there, you've highlighted a square, you've counted the number of cells there, you've written that down, you've made it clear that's the cell you're counting, because I need to be able to give the examiner proof that you know what you're doing, okay? So we need a type C square in each of your 2 photographs. You need a table or 2 tables with numbers of yeast cells in 10 different C squares, all right? And headings, you've got 2 flasks, headings, 10 figures, and at the bottom there, we've got a mean or average. So you will end up with the average or the mean number of yeast cells. So let me just write here. So you might have flask 1. I'm just making numbers out of my head, okay? So say you work out that you've got a mean 7.5 yeast cells per C square. Right, each C square volume, is, well, all right. So if you had 7.5 yeast cells per C square, that means 7.5 cells in whatever that volume is. So you divide your average by whatever that volume is to give you the number of cells per millimeter cubed. So you might get a figure like, let me just get my calculator here. 1.5. So say you got 30,000 cells per millimeter cubed. We want you to write that in standard form. So yesterday we knew standard form is where you move the decimal point. So you've got 3.0. So you move it 1, 2, 3, 4. Okay. You do the same for flask 2. You can do it beside it, you can do it underneath it. I don't really care. Doesn't matter. But eventually you will end up with 2 numbers. And just say that we end up with that figure there for flask 2. Flask 1 only had 1 gram of glucose. Flask 2 had 2 grams of glucose. Why have we got more cells in flask 2? Well, obviously that's why, because there's more food. Now you want to link that to population growth curve. So initially, whenever I put the initial sample of yeast cells there, we would have had a small number, and after a few hours then we would have started to get this exponential growth and then level off. What do we call this here? There's a name for that number. So we're talking about the carrying capacity. We know that if you change the resources there, you change the carrying capacity. So you need to write a few sentences. You need to write a conclusion. You don't need to write a method, or you don't need to write anything else. All you need is your results table, your photographs, your calculations. and then a conclusion. So in your conclusion, you need to state, basically you need to compare the population sizes and quote your figures. Okay, so obviously flask such and such had a larger population than the other. It contained so many cells per whatever, whereas the other flask only contained such and such. This is because, so why you need to say why there's more yeast cells. In other words, why has the population got bigger? Why more yeast cells? Why is the population larger in one flask than the other? So you're talking about the glucose. It would be a good idea to sketch the two graphs. Now, you're not putting any numbers or anything on it. To sketch the two graphs that you would get, showing the difference. Now, I can't do that for you. I can't put it up there. But if you're saying that giving it more sugar, giving it more glucose is going to increase carrying capacity, you just show that on a graph. All right? Now, bear in mind that temperature was the same, everything else was the same. So it's not going to speed up the rate of reproduction. If we'd done it at different temperatures, you might have got faster reproduction. You're going to get the same rate of reproduction, but just the fact that one has more resources than the other. So I'm really talking about 4 or 5 lines and a sketch, all right? Not a whole big page or anything like that. You don't need to write what you did. You don't need to write a method. Simply 2 photographs, table, calculations, and a few lines of a conclusion. Any questions? Okay, so if you can't see 10 squares in your photograph, that's fine. Look at somebody else's, get some of their figures. All right? So if we could get that done for Monday, that would be good.